Okay, any questions from last time? All right. So last time, what were we doing? We talked about all these Boolean functions, attractors, all right? Then the steps to the attractor, right? The levels, right? All that stuff we covered. And now we are going to talk about the coefficient of determination. And this is something that was introduced in one of the papers that we will be discussing, right? I told you there were two papers. Okay, there's another paper that is coming up about, all right, about inferring relationships between different genes, okay, based on measurements of gene expression. And there I will talk about the coefficient of determination in detail, okay. Now here the idea is the following, okay. Let's say you're looking at genes on a microarray, right? And let's say some of the genes, and you're looking at the microarrays across different conditions, okay? And you're observing how see, there might be some genes that whose expressions don't change across all the microarrays, okay? So you cannot infer anything if there are no changes. But then there might be other genes that are going up and down together, right? So there might be some relationship between them, okay? And you want to figure that out based on the gene expression, right? Let's say when gene X goes up, gene Y comes down, okay? Maybe there is an inverse relationship, right? That kind of stuff, right? So by viewing gene status across different conditions, say via microarrays, it is possible to establish relationships between genes which will show variable status across the conditions. If the genes don't change at all, if they don't move at all, you cannot say anything, okay? But if they move up and down together, or one goes up and another goes down, you might be able to infer some connection between these genes, okay? Now, owing to the limited number of replications, because typically, if you look in microarrays, there might be 50 or 100, right, at most, okay? So because your sample size or the data is limited, you're going to assume that the gene expression data are quantized based on some statistical analysis of the raw data, okay? Because if it is quantized, then you need less data to learn the relationships. Now, one way to establish multivariate relationships among genes is to quantify how the estimate for the expression status of a particular target gene can be improved by knowledge of the status of some other predictor genes, right? So in other words, like you have all these genes, right, let's say on the slide, and you want to know, okay, so you want to make a guess about the transcriptional activity of a particular gene, okay? Is that on or off? Now, if observing some other genes gives you a better idea, all right, about predicting whether this particular gene that you're interested in is on or off, right? Then you will say that there is some knowledge in those predictors about this gene, okay? That's what we want to quantify, okay? So this can be quantified via what is called the coefficient of determination, right, or COD, which is defined as follows. It's epsilon O minus epsilon opt over epsilon O, okay? So first, you try to get a predictor of the target gene, all right, without seeing any of the other genes on the slide, okay? So what is the best predictor? It's probably the mean, all right, the sample mean of that gene. That's the best predictor, all right? So for the best predictor, there will be some mistake, right? And that mistake is going to be the variance, right? Right? So basically epsilon 0 is the best prediction that you can make about the activity status of a gene, right? Without seeing any other genes, without any side information, okay? Epsilon opt is the error that you will get for the best predictor that makes use of that side information, the information about those other genes, okay? And this ratio is defined as the COD, right? So you look at extreme scenarios. Suppose, when you're using those extra genes, those predictors, you're not doing any better than what you were doing before. Then epsilon opt in that case would be equal to epsilon O, right? That means the error in the absence of any observation about the predictors, and you would get a COD of 0, right? On the other hand, when you see those other genes, okay, if you are able to predict with 100% accuracy, right, then the error in that case, that epsilon opt, the error that you get seeing the predictors is actually 0. And so you get a COD of 1. Okay, so this is a number between 0 and 1, and it is telling you about how much of extra information are you getting from the predictors, okay? So it's similar to like if you're using bases, okay, if you have like vectors which are dependent on each other, okay, and you're trying to do Gram-Schmidt or something like that and try to get an orthogonal basis, okay, is there any new information? That's the kind of question that you're asking here, okay? And again, I mean, you have to calculate these quantities, right? Epsilon 0 is the error of the best numerical predictor of the target gene in the absence of observation, okay? It's easy to define this, but what is the best numerical predictor of the target gene? So I have to first find the best numerical predictor, then I have to calculate the error associated with that, okay? So there is error estimation involved. Same thing I have to do the best or optimal predictor of the target gene based on the predictor genes. And then I have to calculate the error of that, and then I can get the COD. Okay, and if the COD is high, means there is a good relationship between those genes, right? One may be predictive of the other, right? If it is low, means maybe they are not that closely related. Okay, so that paper that I'm going to discuss on coming up with multivariate relationships between genes based on gene expression microarrays, right? That paper is going to focus on this COD, right? You're going to define a COD, and again, you have to estimate the COD from sample data. Nobody is coming and giving you the probability distribution. It's the same problem like the Bayesian classification problem, right? You don't have all those class conditional probabilities, right? Prior probabilities and so on. You have to estimate those based on the sample data. So same kind of issues will come up over there in that paper too. And that paper I will discuss in detail as soon as I finish this particular chapter. Okay. Maybe I will get started on it on Wednesday and then finish and take it up the following Wednesday, after you've had your break on Friday and then test on Monday, and then, probably the following Wednesday we will try to finish that up, or at least make more progress on it. All right, any questions? So this nonlinear form of the COD, as I've defined here, is essentially a nonlinear multivariate generalization of the familiar goodness of fit measure in linear regression. If you're familiar with linear regression, they have something that is called a goodness of fit measure, right, where you're trying to fit a curve, and so this is a generalization of that. The COD, or the coefficient of determination, measures the degree to which the best estimate for the transcriptional activity of a target gene can be improved using knowledge of the transcriptional activity of some predictor genes relative to the best estimate in the absence of any knowledge of the transcriptional activity of the predictors. Right? So if you don't have any side information, what's the best you can do? Which side information can you do any better? Right? And the extent to which you can do better, that is quantified by the COD. So the COD is a number between 0 and 1, a higher value indicating a tighter relationship. Right? And I already made that argument before. So in the next figure, on the next slide, we show a COD diagram for 3 genes, right? P53 is one of them. You have already encountered that, okay? So don't ask me what is P53. We already covered that in the molecular biology section, right? So P53 is one of them. Then there is P21 also we talked about, right? That's a protein with a mass of 21,000 Daltons. And then there's MDM2. So if you go ahead and, take sample measurements, right? You see, P53, it is activated in response to stress, right? DNA damage stress, right? We talked about that. So, so you get the data basically from some stress response experiments, right? And then you use that data to do these COD calculations, right? So the following figure shows the COD diagram for the target gene is P53 and 2 predictor genes P21 and MDM2, in which the CODs have been estimated in the context of a study involving stress response. Right? So you're subjecting the cells to stress, then you're looking at the expression levels of these different genes, right? And then looking at that data and trying to calculate the best predictor, right? In the absence of observations, then with observations, and then do all the detailed calculations. Okay, so this stuff is repeated in that paper, okay, that I'm going to discuss. So you can take a more detailed look at that later. But the net result is that the COD, right, of P21 predicting P53 is 0.227, right, not very high. Of MDM2 predicting P53 is 0.259, right. But if you use, instead of using just one predictor for predicting P53, if you use the 2 of them together, MDM2 and P21, right, then the COD goes up. It becomes 0.452, right. So we see that the individual CODs for P21 and MDM2 are 0.227 and 0.259 respectively. But when you use them together, the COD for the predictor set increases to 0.452. Right? Now, biologically, it is known that P53 is influential, but not determinative of the upregulation of both P21 and MDM2. Right? If P53 turns on, P21 can be turned on, MDM2 also can be turned on, right, under some conditions, right? And hence, it is not surprising that some level of prediction of P53 should be possible by a combination of these 2 genes, right? And note that the prediction of P53 by P21 and MDM2 results from the regulation of P53 on them, because P53 status affects both these genes. That's why looking at them, you are able to say something about P53, right? Now, going the other way, from the same study, they found that the COD for P53 predicting P21 to be 0.473, right? Now here, one point to note is that when you're increasing the size of the predictor set, you have increased the COD. This was 0.227, 0.259, now it jumped up to 0.452, right? So this is to be expected because, excuse me, increasing the size of the predictor set cannot result in a decrease of the COD. Now, another thing to note is that in that figure, and see, one thing that I should say is that the paper that we will discuss, it has, here I'm highlighting only one relationship, okay? There are thousands of genes, okay? So there are lots of relationships over there, okay? So many of those COD relationships, they are highlighted in that paper, right? And in fact, you already have a copy of that paper. I haven't discussed the paper, you have a copy of that paper, you also have the explanatory material, which I will discuss here, right? So in the figure, furthermore, MDM2 and P21 have got very similar CODs relative to P53, and there is a significant increase when they are used in combination. On the other hand, it's possible that there might be very little, if any, predictability is gained by using predictors in combination. Moreover, you can have a situation where the individual predictors might be very poor. They might have CODs very close or equal to 0. But when you combine them together, then the joint COD is 1, right? Because this is a nonlinear thing. It's similar to like an AND gate, okay? You put in all the different inputs, you're getting output 0. Then when you put in everything together, right? You put in all but 1, you get output 0. Suddenly you put in the last one and then you get an output of 1, okay? So this kind of situation shows that it is risky to assume that a predictor G1 and a target G0 are unrelated because the COD of G1 predicting G0 is very low, right? The situation is akin to that in classification, where you could have a situation where a feature might be poor if it is used by itself, but in combination with other features, it might be really a great feature set to use, right? So the issue in both settings is the danger of marginal analysis, right? Drawing conclusions about variables from marginal relationships instead of joint multivariate relations, okay? And this is what biologists do all the time, like what they call pathway information and all that. It is based on marginal relationships, okay? In one experiment, in some context, they observe something, and these pathways are also being updated all the time, right? There are companies that have pathways that are available on the internet, okay, and these get curated and updated all the time. But again, all said and done, these are based on marginal relationships, right? You have not done comprehensive experiments, taking all the variables into account, right? All right, any questions? Now, starting from this COD information, right, you can actually build up networks. How? For example, let's say you have 10 different genes, okay? So based on the gene expression data, you find out, what are the best predictor sets for each of these genes, right? And then you assign weights to these different predictor sets and, let the network update according to those weights, right? So given a target gene, several predictor sets may provide equally good estimates of its transcriptional activity as measured by the COD. So given one, let's say P53, there might be many different sets of genes that are able to predict P53 with the same COD. So those different predictor sets are equally good. Moreover, you could go ahead and rank several predictor sets via their CODs. You can say that a particular predictor set is better than another one if it has got a higher COD for the same target. Right? So such a ranking provides a quantitative measure to determine the relative ability of each predictor set to improve the estimate of the transcriptional activity of the particular target gene that you're interested in. Now, while attempting to infer intergene relationships, it makes sense to not put all your faith in one predictor set, right? Especially since, the data is supporting several different predictor sets. You could, say that you're not going to just go with one predictor set, right? You're going to use different predictor sets. So instead, for a particular target gene, a better approach is to consider a number of predictor sets with high COD. So all of them are good predictor sets, okay? So now you're going to say that sometimes the network will update according to this predictor set, sometimes according to another one, okay, if you have equally good predictor sets, right? And so you will get, like, instead of a deterministic network, you're going to get something that's going to become stochastic, right? So considering each retained predictor set to be indicative of the transcriptional activity of the target gene with a probability that is proportional to its COD, represents feature selection for gene prediction. Now, one thing I should point out, that this business of gene prediction, this is just like the classification problem, okay? In the classification problem, what are you doing? You're looking at a feature vector, right? And then you're trying to spit out a class label, right? It could be 1, 0, -1, whatever, okay? Here also you're trying to do exactly the same thing, okay? You're looking at the predictors, so that's like a feature vector, right? Look at the expression of the predictors, and you're trying to say whether the gene that you're interested in, the target, is in 1 or in 0. Okay, so this is a classification problem also. It's similar, right? So having inferred, and again, I mean, yeah.
In which case is this practical is useful?
Practical what? I didn't.
I mean, you predict gene expression of a given gene by using real data on expression of other genes, and you know that there's some kind of relation between this gene expression and the gene you're interested in. But when would you when we would use this tool, let's say, or this?
No, see, the biologist may not know, right, that which gene interacts with which one, okay?
Yeah.
So the way that paper has been written, okay, in that paper they have used it on data, okay, to verify known relationships, all right, and also to find out unknown ones, okay. So you may not know that like some gene affects another one, okay, but you look at this data, then you figure. So some of them, and in fact, I think the people that actually provided the data, okay, which is basically Mike Bittner and others who are right here at the center, they even made up fictitious relationships and gave some data, okay. So the person that designed the COD, right, they went through these algorithms. They didn't know that it was a fictitious relationship, but they were able to pick it up. Now the bad thing about the COD is that the COD does not give you any causality, okay. It's like a correlation, right? It doesn't tell you which one is the cause, which one is the effect, okay. That's the problem, right? So if somebody wants to do research, then you would be in better shape, right? I mean, yeah, it doesn't give you any causality. It's just telling you what is going up and down together, that some relationship is there, yeah. All right, any other questions? That's a good question. So, now, having inferred the intergene relationships in some manner, this information can be used to model the evolution of the gene activity profile over time, right? You remember gene activity profile is basically the sets of ones and zeros telling you which gene is on, which one is off at a particular time, okay? So that evolves with time, okay? So now it is unlikely that the determinism of the Boolean network model will be concordant with the data, okay? Because, in the real world, you're going to have stochastic influences, okay? I mean, you're not going to measure everything with 100% accuracy, right? So what you're going to do is, so if it was just the Boolean model, all right, then you probably would just pick the highest COD predictor set, all right, and say that each gene will update at the next time step, all right? And again, the time step also, it's not set in concrete, okay? It's up to you to define. What is the biological time step? Is it 1 second, 1 day, all right, 10 days, 1 year? Okay, you don't know, all right? What is the biological clock, all right? But anyway, assuming that you can agree on the biological clock, this predictor that you get, okay, let's say using the COD, if you use just one predictor set for each gene, then you will get a Boolean network, right? But instead of that, if you say that we are going to have multiple predictors for each gene, okay, and I'm going to say that gene number 1, let's say, has 3 predictors, all right, 3 predictor sets. Gene number 1 will update at the next time point according to the first predictor set with probability 0.1. According to the second predictor set with probability 0.2, according to the third predictor set with probability 0.7, all right? Then this will become a stochastic dynamical system, all right? And it will become a generalization of a Boolean network. It will become what is called a probabilistic Boolean network, right? And I will talk about that in the next few minutes, all right? So one could pick the predictor set with the highest measure of predictability, but as remarked previously in the case of the COD, there are usually a number of almost equally performing predictor sets. And for them, we will have only estimates from the data, okay? So by associating several predictor sets with each target gene, it is not possible to obtain with certainty the transcriptional status of the target gene at the next time point, because one set of predictors might say it's 1, another set might say it's 0, and so on, okay? So you don't know for sure, okay? So you can go and compute the probability that the target gene will be transcriptionally active at time t plus 1 based on the gene activity profile at time t. Activity profile, or actually the entire profile, may not determine the level at time t plus 1. It might be just 2 or 3 genes, okay? 2 or 3 predictors that will determine. So the time evolution of the gene activity profile then will define a stochastic dynamical system. Now again, you're making the assumption that the gene activity profile at a particular time point depends only on the profile at the immediately preceding time point, okay? So then this dynamical system is going to become Markovian, okay? Markovian is like giving your parents, you have to forget about everything else, all the information is there, okay? Immediate predecessor information is good enough, all right? So such systems, these Markovian dynamical systems, can be studied in the established framework of Markov chains and Markov decision processes, okay? And you are from industrial engineering, so it's close to your home turf, right? I mean, operations research and all that stuff, so it's coming, right? The only problem is how to link that to the real world, okay? I mean, who said that it's a first order Markov process, okay? We don't know, right? So these ideas can be mathematically formalized in probabilistic Boolean networks or PBNs, right? And in a PBN, the transcription, so this is a generalization of a BN, right? So in a PBN, the transcriptional activity of each gene at a given time point is a Boolean function of the transcriptional activity of the elements of its predictor sets at the previous time point. And the difference between a PBN and a Boolean network is that in the case of a Boolean network, the predictor function is set in concrete, okay? In the case of a PBN, there will be a number of possible predictors, and one of them will be chosen at each time point. So is that clear to all of you here? I mean, I think is that clear to all of you? I mean, like, I think this relates to whatever you guys are familiar with, like all this stochastic stuff and all that. Yeah, go ahead.
How do you determine which predictors?
Well, I mean, there are different ways, okay? And again, I mean, all this is more theory, okay, than actual this thing. One way to do that is, what you could do is, for each gene, right, you could have a number of predictors, okay, and you have their CODs, okay? So you could assign the probabilities in the ratio of the CODs. The one with the highest COD, okay? So for each predictor, let's say you have, for each gene, you have, let's say, four predictors, okay?
For different sets of predictors.
Yeah. Then for predictor set number 1, okay, let its the probability associated with that getting picked up be its COD divided by the sum of the CODs of all the predictor sets for that gene. That
You would always use the same one because the COD doesn't change.
No. You would not always. You're picking it with a certain probability. The probability is constant, all right, but it doesn't mean that you're always going to pick the one with the highest probability. Yeah, it's a random thing. It's a stochastic thing. So, yeah. So you'll pick one maybe now, or it's like flipping a coin. Okay, if the probability is 1/2, all right, sometimes one will get picked, or sometimes the other will get picked. Yeah. So, yeah, the machinery is all there. I mean, like, it has been worked out several years ago. I worked on some of these things too. Machinery is there. My challenge is how to get it to map to the real world. Okay, and because the biologists, they believe in pathway information, okay, which is marginal. So you can go and give them a big lecture by marginalism, but they are not going to look at all your models, your stochastic models, and buy into that, okay. So how do you bridge the gap? Okay, well, the idea is similar to, if you're in control systems, okay, there are people who design all kinds of sophisticated controllers using very elegant mathematical theories, okay. But guess what runs most of the controllers in the real world? It's PID, okay, proportional integral derivative controllers. So motivated by that, many years ago, we tried to, design optimal controllers with the PID constraint. So here also you have to come up with networks that have the pathway constraint built into them. Because if it's anything different from pathways, you say, I'd build something from scratch, they're not interested, okay. And pathway, although it's marginal information, right, I think it's worth something, right, because, they have observed across many experiments. It may not be 100% correct. They have observed across, so you benefit from that, okay. It's similar to like you're getting a PhD, okay, but if your parents tell you something, they might have bachelor's degree, okay, but they know something, okay, like that might be the pathway information, okay. And so you cannot say, I'm a PhD, I know everything about, everything there is to know, I'm not going to listen to anybody. You have to combine both of them together, okay. Prior information and, did you get the? So here also it's similar. You cannot just say that you biologists, it's all marginal. Okay, I'm not. This is no good. Okay, this is all nonsense. Okay, you cannot say that. Okay, because they have been in business. They are treating diseases. They are doing things like that. So it's not that engineering is the only thing that works, right? So I think it's a middle ground, but I don't know where exactly you draw the thing. But if the biological data could be made to fit this framework, all the theories, heavy duty machinery is already there, okay. It is there in the controls operations research literature. A lot of smart people have beaten a lot of these problems to death, right? And you probably know about that, right, from industrial engineering. Yeah. So as I said, for instance, when using the COD, the choice of the Boolean function and predictor set can depend on the COD based selection probabilities associated with the different predictor sets. So this kind of probabilistic generalization of a Boolean network, in which the Boolean function is randomly selected at each time point, okay. At each time point, the predictors are selected with certain probability for predictor sets for each of the genes. This is called an instantaneously random PBN, okay. But this might have too much of randomness in it, because what the biologists believe, right, is that maybe you have a Boolean network, right? It's running for a while, and then suddenly in response to some external stimulus, the network flips, the network changes. Okay, so you really don't need to be changing the network function or the predictor function at every time step. Okay, but there should be some possibility for change. Okay, so that gives you what is called a context-sensitive probabilistic Boolean network, and that's what I will talk about next. And all of these, we have worked on these, and we've worked out all the control and things like that, right? How the dynamics evolve and all that. But again, until you connect up to the real world, all that is theory. Okay, so that's the big challenge. So instead of simply assigning Boolean functions at each time point, one could take the perspective that the data come from distinct sources, each representing a context of the cell. Okay, so in a particular context of the cell, it's a Figs Boolean network that is determining the dynamics of the cell, okay, or the phenotype. So from this viewpoint, the data derived from a family of deterministic networks, and were we able to separate the samples according to the context from which they have been derived, then there would in fact be CODs with value 1, indicating deterministic biochemical activity for the wiring of a particular constituent network. So under this perspective, the only reason that it is not possible to find predictor sets with COD equal or very close to 1 is because they represent averages across the various cellular contexts. Okay, so you have a context-sensitive probabilistic Boolean network. So this perspective, and again, I mean, those of you that are interested in reading papers, what I'm presenting here, those are documented in papers. Okay, there are papers published about 10, 12 years ago, and each one of them, I mean, if you have instantaneously random, that's the first paper. Then there are context sensitive. Also, there are papers that have been published. Okay, and each one, I'm not asking you to present those. If you want, you can, but each one of them would be interesting reading. So this perspective results in the view that a PBN is a collection of Boolean networks in which one constituent network governs gene activity for a random period of time, right, before another randomly chosen constituent network takes over, possibly in response to some random event such as an external stimulus or genes not included in the model network. See, remember, when you're building this network, right, you cannot include all the genes, so there will be latent variables, right? So every time you will not see the same transition, even when the input appears to be the same, because there are some hidden variables that have been left out, right? So since the latter is not part of the model, this network switching appears to be random, right? So this model defines what is called a context-sensitive PBN, right? And the probabilistic nature of the constituent choice reflects the fact that the system is open, not closed, right? So we don't know, I mean, we don't have the system modeled 100%. Even if you're doing deterministic modeling, you can never model 100%, right? You'll have some modeling errors. So the idea is that network changes result from the genes responding to latent variables external to the model network. And the context-sensitive model will reduce to the instantaneously random model by allowing the network to switch at every time point. Right? If it's context-sensitive, means for some time it's just a fixed network, right? And then it switches, right? But if you want allow switching at every time point, right? Then again, from those predictor sets that you have, you're switching at every time point. It's like you're tossing a coin and like switching to some network, right? And in that case, it becomes instantaneously random. So every time point there is a switching. Now, much of the theory and application of PBNs, because PBNs are binary, right? 1 and 0, the two states, applies directly to the more general case, which need not possess binary quantization, and which are also called PBNs, owing to the multi-valued logical nature of functional relations for finite quantization. For example, you have ternary. We have seen in the microarray case, okay, when you look at the data, red over green significantly greater than 1, red over green almost 1, red over green significantly less than 1. So that's ternary, okay. We would still call them probabilistic Boolean networks, and the same kind of theory carries over even when you have this multi-valued logic, right? So a particularly important case is ternary quantization, where expression levels take on the values +1 for upregulated, -1 downregulated, and 0 invariant, right? So, and again, now this is a mathematical definition of a PBN. Okay, so a PBN is composed of a set of n genes, x1 through xn, each taking values in a finite set V. We have assumed that it's only 0 and 1, containing d values. So for us, d is 2, right? And a set of vector-valued network functions, f1, f2, up to fr, governing the state transitions of the genes. Okay, so there are different network functions depending on which network is selected. So to every node xi, there corresponds a set f, or fi, which is made up of Li different predictor functions, right? Where each Fij is a possible function which is called a predictor determining the value of gene Xi, and Li is the number of possible functions that are assigned to gene Xi. So this is just the mathematical part, like, I mean, the ideas or whatever I have already explained, but we can just quickly run through this. So each network function will be of this form, right? So you would have selected a predictor for each of these genes, right? F1, K1, F2, K2. So among all the predictors for gene 1, you selected one of them. All the predictors for gene 2, you selected one of them, and then you got the set of all the predictors for the different genes in the network, right? So each vector function Fk, which maps basically 0, 1, all right, n, right, to 0, 1, n, acts as a transition function or mapping representing a possible realization of the entire PBN. So thus, given the value of all the genes, the gene activity profile x1 through xn, Fk evaluated at x1, x2 up to xn is giving you the state of the genes after one step of the network given by the realization Fk, right? And the choice of which network function Fj to apply is governed by a selection procedure, right? So at each time point, a random decision is made as to whether to switch the network function for the next transition, with the probability Q of a change being a system parameter, right? So maybe you flip a coin. Should you switch or shouldn't you? Okay. If you don't switch, you just stay with the same network function. If you switch, then you will choose one according to the probability distribution across the different predictors for a gene. So if the decision is made to change the network, and again, if somebody is not following something here, right, just stop me. We can discuss further, right? Because this is again stuff that is distilled from those papers, right? Which you can go and read. But I think we are trying to give the big picture and cover stuff in an intuitive way. So there's no reason that you shouldn't understand. And if you don't, just question me, and we'll stop and try to explain a little bit more. So if a decision is made to change the network function, then a new function is going to be chosen from all those R network functions, with the probability of choosing Fk being the selection probability Ck. Okay. And this is close to your home turf because most of you have got background in stochastic processes. Okay, I mean that's what you're trained in. Okay, the biology and all that is kind of outside your comfort zone. But my advice to you would be to learn more about the biology. Okay, because that's where you can make a mark, right? That's where you can build up the connections. All the stochastic, stuff and all that, it's meaningless if nobody uses it. Okay, if it cannot be used anywhere. Like some of you are on agri-life projects, right? You should look at their projects and try to understand what the biological problem is. Okay, and then try to see if you can find some theory instead of picking up some theory. I like Viterbi algorithm. I'm just going to go and apply it no matter what. Okay, I'm going to force the plant or the water use project and just apply that. That's not going to get you anywhere. Or I don't know, if you're doing in cancer research also, try to learn the problem, try to learn what will be important to them. Okay, what is going to be a high impact thing, right, for them. No, yeah, for you the high impact is just to get your degree. Okay, that's okay. I'm not saying that you spend your life, not graduate. Just dedicate yourself to the cause. I'm not saying that. Okay, but don't run around, with a hammer looking for a nail. Okay, because first maybe it's a screw. You need a screwdriver. Okay, or something like that. Right. So first try to understand their viewpoint, because if you can do that, you'll be pretty well received. Okay, because for most of the people like engineers and math people, they don't like to think outside the box, and they don't want to get out of their comfort zone. And same way the biologists also will not want to get out of their comfort zone, right? The pathways or whatever is drilled in their heads, because, and you need their help, because the problem is our theories are great, but if you're going to link it to the real world, you need data, okay? And you don't have enough data to learn the network, okay? Which means that you have to use some prior information, right? Some information from them, to constrain the problem, to reduce the size of the problem. So you guys are in a unique situation. You have strong math skills and all that, and you can put that to work. Learn all this stuff. Do your own thing. But again, see, if you're interested just in the theory thing, you should be doing your PhD in controls, communications, and all that. There are open hard problems left in a lot of these areas. Okay, and you can spend and get a kick out of solving that hard problem. Okay, but it's not going to have that much of an impact, right? But if you're looking for impact, and in this interdisciplinary area, you're going to have to spend the time to study the biology, right? And not just what I covered, right? See, that's another problem. A lot of students I found, they didn't learn any biology beyond what I covered in this class, okay? What I covered in this class is like the ABC, okay? If you're going to delve deeper into some other problem, You need to become like the world's leading expert in that, okay? And then you apply your mathematics and all that, it's going to be very cool, all right? Do your experiments and show that things work, right? Because things have to be taken to the finish line, right? Like if you, let's say you're working on, treating cancer, for example, right? Nobody cares how hard you worked, okay? How many sleepless nights you spent, right? If you can find some drug, all right, that works wonders, all right, you've made it, right? So you have to take it to the finish line, right? And taking to the finish line is not just taking my tests, okay, and then leaving it at that, okay? I'm saying that you'll know enough biology to just get by, right? But you need to build up pretty deep domain knowledge, and you don't have to build it up for all of biology. Nobody's asking you to become the world's leading, molecular biologist, come engineer, right? No, you focus on the particular problem, pick your biologist and work on that. So if a decision is made to change the network function, then a new function is chosen from among F1, F2 up to Fr, with the probability of choosing Fk being the selection probability Ck. Now let F given by F1, F2 up to Fn be a random vector taking values in F1 cross F2 up to Fn, right? So that is F can take on all possible realizations of the PBN. Then the probability that the predictor Fij is used to predict the ith gene, right? Where J varies between 1 to Li because the ith gene has got Li predictors, right? That will be equal to basically the probability over all the networks, right? For which the ith gene is predicted by the jth predictor, right? And again, for understanding this a little bit better, you might want to just read the paper, okay? It is basically bookkeeping in probability, right? Probability and statistics in basic probability, how you're trying to compute the probability of a compound event from elementary events and all that is basically that, okay? So since theCij's are probabilities, they will have to satisfy this relationship because, for the ith gene you have Li predictors, right? And, the value has to be according to one of the predictors, okay? So this has to sum to 1, right? So no deep rocket science there, right? Now it is not necessary that the selection of Boolean functions composing a specific network be independent because you have Boolean functions for each gene. They don't have to be independent, okay? So it is not necessarily the case that the probability that the ith gene has got the jth predictor, the lth gene has got the kth predictor, right? Is the product of these individual selection probabilities. That's not needed, right? So PBN is said to be independent if the random variables F1, F2 up to Fn are independent. So in the dependent case, product expansions such as the one given in the above equation, as well as ones involving more functions, they will require conditional probabilities. Okay, no big deal. Okay, we know if it's independent, right? The conditional probability is the same as the original probability, but if they're dependent, if the probability of intersection of two events will be basically written down in terms of conditional probabilities. So if the PBN is independent, then there are L, capital L, which is the product of the number of predictors, right? For each gene, for the ith gene, you have Li predictors, right? For all the genes together, you'll have this many realizations or constituent Boolean networks, right? So moreover, for an independent PBN, if the kth network is obtained by selecting FI IR for this gene, right? Then the selection probability is given by the product of these individual selection probabilities. Okay, and again, I mean, I'm just going through these, this PowerPoint presentation here, but this is better understood if you just look at the paper, look at the equations. Okay, this is a 2002 paper, and then work out the math. Okay, nothing deep over there, and I'm sure you'll be comfortable with it. Now, in addition to, so we talked about Boolean networks, then probabilistic Boolean networks, both instantaneously random, right? And we also talked about context-sensitive Boolean networks, right? In addition to that, another class of PBNs was introduced, a PBN with perturbation, right? Where you say that some gene might just randomly flip. In addition to the modeling, right? Maybe there is a mutation, some gene has changed from 1 to 0, 0 to 1. So you're allowing probability of that, okay? So PBN with perturbation can be defined by there being a probability P of any gene changing its value uniformly randomly. Because you could have multi-valued genes, okay? So if it is binary valued, there's a probability of half that, a small probability that it could change between one of those two values, right? If there are, 10 values, then, there'll be a small probability, a uniform probability, right? Of changing to any of those other 9 ones, okay? So you have two things here then. You have a network could switch, okay, you could also have a gene flipping, right? So a network switch corresponds to a change in a latent variable causing a structural change in the functions governing the network. For example, in the case of a gene outside the network model that participates in the regulation of a gene in the model. Some other gene outside changed, because of that the network function changed, right? A random perturbation, on the other hand, is a one-time thing, right? It corresponds to a transient value flip that leaves the network wiring unchanged, as is the case of activation or inactivation owing to external stimuli such as mutagens, heat stress, and so on. Okay, they could cause a change. Okay, I mean you apply heat, so something got turned on. Okay, maybe heat shock protein got turned on. Right, that became one. So that kind of thing. Right now, this. See, when you put in a random perturbation, right, it actually has some beneficial effect, but I don't know how realistic it is from a biological point of view. Mathematically, it's a beneficial effect because, if you allow random perturbation, then every state, right, in the Markov chain will communicate with every other state, right? So you have what is called an ergodic Markov chain, right, and then, there will be what is called a steady state distribution, and you can do a lot of elegant math, okay, but from what the biologist tells me, that's probably not a very good assumption, because once a cell goes into, let's say apoptosis or something like that, it's not going to switch, it is there. Okay, so anyway, the state space S of the network, together with the set of network functions, in conjunction with the transitions between the states and network functions, determine a Markov chain. All right, and the Markov chain in this case, see, in the beginning, if you didn't have any if you had instantaneously random Boolean network, right, probabilistic Boolean network, that at every time point you are selecting new predictors, okay. So in that case, the states would be just the gene activity profile, right. They would be the state, all right, the components of the gene activity profile. In the case where you're choosing a network also at some point in time, right, you're going to have states of this form. It'll be the actual state space and the predictor function, right. So it's bigger than the original chain, okay. So if you have a random perturbation, then the Markov chain is also ergodic, right? Meaning that it has the possibility of reaching any state from any other state. And once a Markov chain is ergodic, then it has what is called a stationary distribution, right? And that'll become a steady state. I'm sorry, it'll have a steady state distribution, right? So in the special case, when Q is 1, right, that means the probability of the network function changing at every time point is 1. That means at every time point you're basically going through the list of predictors and choosing, you have an instantaneously random PBN, and in that case the Markov chain consists only of the PBN states, because you don't have to worry about this other state Fj, because at every time point you're choosing like one of the constituent networks. Now for a PBN, the characterization of its long run behavior is described via the Markov chain that it defines, okay, because that is a dynamical system, right? X at time k plus 1 is some Boolean function of the gene activity profile at time k. So an instantaneously random PBN has got equivalence classes of communicating states analogous to the basins of attraction for Boolean networks. If there is perturbation, then the Markov chain becomes ergodic, right, which will guarantee the existence of a global steady state distribution. So in general, whether the PBN is instantaneously random or context sensitive, by definitions, its attractors consists of the attractors of its constituent Boolean networks, right? Now, two events can remove a network from an attractor cycle C. You could have a perturbation, a gene perturbation, right? So it was in some attractor cycle. Now the state looks different, so it's gone from there. Or you could have a network switch. The network has changed, so the attractor landscape has changed. Okay, so you might be in the original state, but it's not an attractor anymore. Okay, so the dynamic equations are now going to be altered. So a perturbation can send it to a different state, and assuming the constituent network remains unchanged and there are no further perturbations for a sufficient time, then it will return to C if the perturbation leaves it in the basin of C. Right? C is the attractor cycle, or it will transition to a different attractor cycle of the same constituent network if the perturbation sends it to a different basin. Similarly, you could have a network switch which could put it in the basin of an attractor cycle for a new constituent network, right? And it will transition to the attractor cycle for that basin so long as the constituent network remains unchanged and there are no further perturbations for a sufficient time. So you can have two possible changes here. Either the network could change, right? Or you could have a gene flipping, right? So if the network changes, even if you don't change your state, now this state is going to have different properties. It may have been an attractor before, it's not an attractor anymore, right? So it's going to move, right? Similarly, if one particular gene flips, the gene activity profile is different. It might have been in an attractor, now it's somewhere else. So it's going to evolve according to the new dynamics. See, whereas the attractor cycles of a Boolean network are mutually disjoint, right? Because because the attractor means you get in there, you stay there. Okay? You cannot simultaneously get to two different attractor cycles. Okay? In the case of a Boolean network, that cannot happen. But in the case of a PBN, there can be intersection because it's only with a certain probability. It's not for sure. Okay, so it could visit different attractor cycles at the same time with different probabilities, right? So the attractors of a PBN can intersect because different cycles can correspond to different constituent Boolean networks. So assuming that the switching and the perturbation probabilities are very small, right? Because these are things you don't want them to happen all the time. Okay, the network switching, it should happen only sometimes, all right? It shouldn't be happening all the time. So the probability of the network switching should be small, all right? Then even the perturbation you don't want all the time. So the perturbation probability will also be small. So if you make that assumption, then a PBN will spend most of its time in its attractors, right? Because it's not just jumping across different states just because the network switched or a gene flipped. Now, I will be discussing, like in the next chapter, right, when we talk about probabilistic Boolean networks with intervention and all that, I will give you a simple example to clarify all these different concepts, right? Like intervention, what does it mean for the gene to flip and things like that, okay? So, and how you could make those questions about whether a gene is flipping or not become important in the context of, let's say, treating a disease, right? But if you define a PBN, right, now you have to infer the PBN from the data, right? So there's the issue of network inference, right? So you confront three impediments in designing genetic regulatory networks from experimental data. Because the network I have presented, all that theory is fine, right? But really, if it's going to be applied to biological applications, you have to infer it from the data, okay? So there are two or three issues that come out. The first one is model complexity, okay? How complicated is the model going to be, right, of the network? You don't have a whole lot of data, right? And the third thing is that you do not have time course data to model dynamics, right? In dynamical systems, in engineering systems, typically you do time domain identification. You excite a system with a sine wave, or you give it a step input, look at the response, then you try to identify, okay? In biology, usually you do not have time course data, okay? The only kind of time course data that they talk about is like maybe they have 200 rats or mice, okay? They kill some of them at different points in time, okay? And that is time course data, okay? It is not data across the same animal, okay? So, I mean, but here in Mike Bittner's lab, he has tagged live cells, okay, with some promoters. He's tagged the promoter region of a gene using a fluorescent protein. So every time the gene is expressed, right, and there's transcription, translation, that light is produced, and he has people doing the image processing who can capture that image and do the analysis. So we can, without killing the cell, we can monitor the activity or the response of the cell to different drugs, okay? It can be done in real time, okay? So we do have the ability to do that. But in general, when the biologists are talking about time course data, it just means that, some of the animals have been killed after 1 month, some after 2 months, right? But the animals that got killed after 1 month, they're not time course data anymore, they're gone, right? So that's the kind of data that you have. But, it's their choice, and their experiments are expensive and all that, it's not easy for them to generate time course data, right? And the rats might die also. So their experiments, see, if we write a proposal, it's like we put the theory and all that stuff and it's done, okay? Their experiment, let's say they started out with, let's say, 200 rats, okay? And they've given them different drugs. There's a control group and then there is a treatment group and all that. What happens if these fellows die, okay? You cannot say that you blew up $2 million, okay, and you produce nothing, right? So you have to have a backup plan, okay? If some of these die, it's very unlikely that, all of them will die, but mix and match so that you produce some data, okay? Because you're getting funding to the tune of millions of dollars, right? And then you cannot go back and say, oh, okay, all the rats died, so I don't have any report to submit, okay? But that's a possibility over there. Yeah, in engineering, of course, you cannot say that unless you yourself died or something like that, So there's lack of appropriate time course data to model dynamics. So numerous approaches to the network inference problem have been proposed in the literature, many based on gene expression microarray data. Here we briefly outline some of the proposed methods for PBNs and the rationale behind each of them. And again, this business of network inference, okay, it's only that the data is coming from the microarrays, okay, or from the biological experiments. But the methods are very much what engineers already had, okay. These must be adaptations of those, right? like adapted to the situation where the amount of data is limited and so on. Okay, so I think let's stop here for today. I will continue from here next time. And no class this Friday and test on Monday. Okay.